This is the Chapter 13 Carb Metabolism, Part 2 commentary. In this first part of the commentary, we're going to go ahead and continue talking about the Krebs cycle and the three main points of regulating the Krebs cycle. I should point out the alternate name for the Krebs cycle is citric acid cycle. So we always have two names for the same thing, so the alternate name for the Krebs is citric acid. We're going to go ahead and focus on these key enzymes in the citric acid cycle because they're responsible for regulation. So citrate synthetase is used in the first step, producing citrate, and then these are the modulators. In the third step, we have isocitrate dehydrogenase. These are the modulators, and then alpha-ketoglutarate dehydrogenase enzyme complex, and all of these are the modulators. Now, going to this slide right here, it's not in your regular slides. I just included it just to give you the big picture. Here is the Krebs cycle, and its ultimate goal is to go ahead and produce ATP. Now, we haven't talked about how it produces most of its ATP yet. So you just take it at face value that ultimately it's going to produce ATP. So what happens if we're in a scenario where we have already have all the ATP that we want? Well, in that case, there's no reason to make more. And again, the body doesn't want to be wasteful. So if we already have a whole bunch of ATP, then we're going to go ahead and slow down the Krebs cycle. On the other hand, what happens if we have very low ATP? Then we're going to need to go ahead and speed up the Krebs cycle. So, going straight to slide 3, which shows the summary, these serve as modulators which help to slow down or speed up those enzymes. Now, it's your responsibility to memorize, aka put on your cheat sheet, all of these structures, modulators, enzymes, which you should have already done in the Chapter 13 Part 1. What I'm going to go ahead and focus on right now is try to show you a pattern. If I have a whole bunch of high ATP to begin with, there's no reason to make more ATP. So, it turns out that as ATP is floating around, it can act as an inhibitor. So when it bumps into, for example, the citrate synthetase found in the first step, it will combine with citrate synthetase, inhibiting it. Now, it's assumed that if you have high amounts of ATP, then you should have plenty of NADH floating around, which also serves as an inhibitor. So we see this for our step 3. For isocitrate dehydrogenase, and we also see it for alpha-ketoglutarate dehydrogenase as well. As these are in high amounts when you have high ATP present. On the other hand, what happens if we have very low amounts of ATP? Well, we need to go ahead and we need to speed up the Krebs cycle so that way it can indirectly produce more ATP. So it turns out that if we have low ATP, well, that means we should have high amounts of ADP. So these are the used up batteries that have already been phosphorylated. They lost their extra phosphate, so they're ADP. Consequently, when they combine with the enzymes they're going to go ahead and act as activators, and they help speed up the Krebs cycle. So know those relationships. Let's go ahead and move on. This is the electron transport chain that we see here and we see here. So we're going to assume that the Krebs cycle, which occurs inside the matrix of the mitochondria, its ultimate goal is to produce regenerated NADH, as well as FADH2. Now in this picture right here, we broke up into two separate electron transport chains. Usually they're together, but we broke them up into two separate electron transport chains. I'm just going to go ahead and focus on the one on top, and then you can go ahead and apply the same principles for the one on the bottom. Focusing on the one on top, here we have NADH. So NAD has a covalent bond and a hydrogen that's attached. When it's exposed to the electron transport chain, it's going to go ahead and give off its covalent bond and hydrogen. The covalent bond is going to is basically made up of electrons. So it's going to turn on what we call metalloproteins within the electron transport chain. And it passes through each of the metalloproteins, kind of like electricity passes through a circuit, until it's grounded. It's ultimately grounded by the electrons passing between hydrogen and oxygen, and they form brand new covalent bonds between hydrogen and oxygen, resulting in new water form. Now, the other thing that NADH gives off, other than the covalent bond electron pairs, is hydrogens. The hydrogen ions, when exposed to an electron transport chain, if the electrons are running through it, basically the metalloproteins act as hydrogen pumps, which serve to pump the hydrogen ions into the intermembrane space. So as the electrons pass through the metalloproteins, each of the metalloproteins, their function is turned on, and they pump hydrogens through. This creates a high hydrogen ion concentration in the intermembrane space. So, in the next part, you have what's called the ATP synthase. Because there's a high hydrogen ion concentration on the intermembrane space side versus the low hydrogen ion concentration found within the matrix, the hydrogen ions naturally pass through this special protein called the ATP synthase. As they pass through, it's kind of like water passing through a water wheel. They go ahead and start turning and powering on the ATP synthase, which serves to basically add on available phosphate ions, PO43-, which is summarized as a P, which go ahead and they put on the phosphates back onto the ADP, forming ATP. So the general reaction is ADP. The used up batteries, they add on the phosphate, forming ATP. So this is the high energy, fully charged batteries. This process is called oxidative phosphorylation. That's the action of putting the phosphate back on the ADP, forming ATP. So, the electron transport chain, and then the passage of hydrogen ions through the synthase, resulting in oxidative phosphorylation of ADP to ATP. We all summarize these steps as the chemiosmotic hypothesis. So the next several slides summarize what I just talked about. In this case, we have the electron transport chain. The electron transport chain is powered by NADH and FADH2. The pairs of electrons power the metalloproteins, aka cytochromes, aka, well, some people consider them enzymes. So I won't. The book considers them enzymes. Good enough. Let's move on. And these metalloproteins are basically hydrogen pumps, which pump hydrogens through. Now, the electrons are then, as they turn on the proteins, they eventually pass back into the matrix, and then you do have low amounts of hydrogen ions floating around the matrix, and these pairs of electrons act as covalent bonds between these hydrogen ions and incoming oxygen gas, which forms water molecules. Here's a nice picture summarizing the electron transport chain. Here we've stuck together both the NAD and FADH2 metalloproteins all together. And as they donate their electron pairs through, they turn on the metalloproteins again. These metalloproteins serve as hydrogen pumps, forming high amounts of hydrogen ions in the intermembrane space. Over here, you see the electrons forming covalent bonds between oxygen with new water. The used up NAD, FAD is recycled back into the citric acid cycle to be reduced again back into NADH, FADH2. So we talked about these three steps which make up the chemismatic process. And the last one, hydrogen ions flow back into the ATP synthase. The alternate name for ATP synthase is F1 ATPase. Yes, you need to know all of this, K and OW. You need to know all of this. All of this should be on your cheat sheet. The ATP synthase basically puts a phosphate back on ADP, forming ATP. This process of putting phosphate back on we call oxidative phosphorylation. So know all of this. Here's another picture summarizing it. Electron transport chain, hydrogen ions passing through, oxidative phosphorylation. And that summarizes this portion of the slides.